What is Methylobacterium ashmali? Do you know anything about it? Does it ring any bells? If not, then don't worry. We are here to tell you all about it. Watch this video till the end to know about it. The genus Methylobacterium contains more species than any other genera within the family Methylobacteriaceae, order Rhizobiales, and class Alpha Proteobacteria. Methylobacterium species are gram-negative, rod-shaped bacteria. The genus was first proposed by Pat et al. In 1976, with Methylobacterium organophilic as the type species. The genus Methylobacterium was first amended to include facultative metylotrophs that have the ability to grow on methane or methanol as the source of carbon and energy, in addition to sugars and organic acids. Another taxonomic study classified all other previously known pink pigmented facultative metallotrophic bacteria under the genus Methylobacterium, Green and Bousfield 1982. Thereafter, 11 species from the genus Methylobacterium were redefined into a new genus, proposed as Methylorubrum, based on 16S rRNA gene sequence, multi-locus sequence analysis, genomic and phenotypic data. Green and Ardley 2018. The genus Methylobacterium consists of 45 recognized species, which are ubiquitously present in a wide variety of habitats, including air, soil, freshwater, and sediments, and can exist either in free form or associated with plant tissues. Methylobacterium species are involved in nitrogen fixation, phosphate solubilization, abiotic stress tolerance, plant growth promotion, and biocontrol activity against plant pathogens. For instance, a novel Methylobacterium sp2a was observed to result in a higher density of lateral roots in inoculated potato crops, even under salt stress conditions. Compared with control plants that were not inoculated with the bacteria, it was also found to exhibit biocontrol activity against several plant pathogens. Furthermore, genomic analysis of Methylobacterium sp2a revealed the presence of metabolic pathways involved in plant growth promotion, including the genes for producing an auxin, 3-indole acetic acid. In an ongoing microbial tracking experiment on the International Space Station, 4 strains belonging to the family Methylobacteriaceae were isolated. Some of the Methylobacterium species that are phylogenetically related to these ISS strains have been isolated from plant sources, indicating that the ISS strains might also display properties related to plant growth promotion. The objectives of this study were to generate whole genome sequences and define the phylogenetic novelty of the ISS Methylobacterium strains using polyphasic taxonomic analysis. The WGS generated and annotated in this study was used to predict biotechnologically useful genetic determinants. Materials and methods. Sample collection and isolation of bacteria. Several surface samples, 1 meter square, were collected from the ISS during microbial tracking 1 flight experiments from 2015 to 2016. Sample collection, processing, and isolation of cultivable microorganisms were published elsewhere. Briefly, the polyester wipes used to collect samples and particulates associated with the sample devices were transported to Earth before being disassociated into sterile phosphate buffered saline solution and plated onto R2A agar medium. The microbial cultures that were grown at 25 degrees Celsius for 7 days were picked from the R2A plates, purified, and further stored for analysis. Distinct colonies isolated from 3 different locations and from a high-efficiency particulate arrestance filter were characterized during this study. These colonies exhibited unique coloration and differential genomic phylogeny. The type strain IF7SW-B2T was isolated during Flight 1, March 2015 at location number 7, the overhead 3-panel surface of the Material Science Research Rack 1, which is used for basic materials research in the microgravity environment of the ISS. The second strain, IIF1SW-B5, was isolated during Flight 2, May 2015 at location number 1, the port panel of the cupola. The cupola is a small module devoted to the observation of operations outside the ISS, such as robotic activities, spacecraft approaches, and extravehicular activities. The third strain, IIF4SW-B5, was isolated during Flight 2, May 2015 at location number 4, the surface of the dining table. Even though the main function of the table was for dining, crew members also used the table for experimental work. The fourth strain was I1R3, isolated from the ISS HEPA filter that was returned aboard STS-134 ULF6 in May 2011 and archived as reported earlier. DNA extraction and whole genome sequences analysis. A biomass of approximately 1 microgram wet weight was collected for DNA extraction from each strain after growing on R2A medium at 25 degrees Celsius for 3 days. Total nucleic acid extraction was carried out using Zemo Biomics 96 MagB DNA kit. Lysis tubes, Zemo Research, United States, after bead beating with a Burton Procellus homogenizer. This was followed by library preparation using the Illumina Nextera Flex protocol as per Illumina document number 100000025416V07. The initial amount of DNA for library preparation was quantified, and 5 to 12 cycles of polymerase chain reaction amplification were carried out to normalize the output depending on the input DNA concentration. The amplified genomic DNA fragments were indexed and pooled in 384-plex configuration. Whole genome shotgun sequencing was performed on a Novasec 6000 S4 flow cell PE 2x150 platform with a paired end module. The data were filtered with NGS QC Toolkit V2.3 Patel and Jain 2012 for high-quality vector and adapter-free reads for genome assembly. Cutoff read length for HQ 80%. Cutoff quality score 20. The number of filtered reads obtained were used for assembly with SP8 3.14.0 genome assembler using default parameters. The genome was annotated using the National Center for Biotechnology Information Prokaryotic Genome Annotation Pipeline 4.11. In addition, functional annotation of genome and seed categories were assigned to the genome by implementing the rapid annotations using Subsystems Technology tool. Genomes of all other strains used in this study were downloaded from NCBI, and the genomic relatedness of ISS strains was identified based on average nucleotide identity calculations and digital DNA-DNA hybridization analysis. Fast ANI was run on all the genomes using the default parameters. MashMap identity cutoff IO is 80%, non-overlapping fragments of size I 3 kb, and minimum count of reciprocal mappings 50. Phylogenetic analysis. Phylogenetic analysis was carried out based on 16S rRNA gene sequences and MLSA using 6 housekeeping genes: ATP synthase F1 beta subunit, DNA strand exchange and recombination gene, chaperone gene, DNA-directed RNA polymerase subunit beta, glutamine synthetase type 1, and DNA gyrase subunit B for differentiating Methylobacterium species. The 16S rRNA gene sequences of type strains of all 45 Methylobacterium species were included in the phylogenetic analysis. In addition, representative species of genus Methlorubrum, Enterovirga, Microvirga, and Neomegalonema from family Methlobacteriaceae, Rhizobium from order Rhizobioles, Caulobacter from order Caulobacteriales, in class Alpha Proteobacteria were included. Pseudomonas aeruginosa was selected as the outgroup. The 16S rRNA gene sequences of all strains were retrieved from the NCBI, except for the 4 ISS strains, which were recovered from their respective WGS. Phylogenetic analysis based on housekeeping genes and MLSA was carried out with type strains of 24 Methylobacterium species and representative species of other genera. All the gene sequences were retrieved from the genome sequences using RAST v2.01. The individual gene sequences for all strains were aligned separately using Clustal W, and then the maximum likelihood tree was generated using Mega 7.0.26. For MLSA, 6 housekeeping gene sequences for each strain were concatenated manually and aligned using Clustal W, and then the maximum likelihood tree was generated using Mega 7.0.26. The genome-based tree for the Methylobacterium species, including ISS strains and representative species of other genus with available WGS, was constructed using G2 tree. That's it for the video. If you enjoyed it, kindly like the video and subscribe to our channel.